This video is Unit 2, Learning Target 9, Macromolecules. This will represent some of the more difficult things in this unit. Just to start off, I'm going to list some key elements that are found in living things. All living things are primarily composed of these 6 elements. That doesn't mean that there aren't other elements inside living things, but these are the primary elements found in living things. And you'll notice they make a handy acronym, Starch. I don't know what that is. Perhaps you can find another word, but that works for me. And they're all notice periodic table elements, and they all have the solo letter for the abbreviation. So what is a macromolecule? A macromolecule is a large molecule. Macro means large, as opposed to micro, which means small. Macromolecules are large molecules. They're in 4 classes, found in 4 classes in living things: carbohydrates, lipids, proteins, and nucleic acids. I like that picture. Macromolecules are made up of individual units called monomers. And a monomer is just a single unit of something. In this case, we're talking about macromolecules. And so multiple monomers can join together to form a polymer. So here you have multiple Legos to make the White City, and looks like Isengard back there, and of course the army that's invading. All right. Now, a couple of reactions that we need to be familiar with. When 2 monomers are joined together, that reaction is called a dehydration synthesis. So what do we mean by that? Well, let's look at these 2 words. Dehydration, what does that mean? That means to pull water out of. So we're dehydrating something, we're taking water out. And the word synthesis, you should learn this word, means to make something. To make something. And so we're going to make something by pulling water out. And so here's an example right here below. You see one glucose molecule, which is a monomer of sugar. We see a fructose molecule, which is another monomer of sugar. In order to join them together, we pull a water out of that molecule, thus dehydrating it, and it joins them together. You have this covalently bonded molecule there at the bottom. And this is, as it were, sucrose. This is like table sugar. So that's when you're joining them together. So how do you break them apart? Well, let's look at this word. Hydrolysis is how you pronounce that. First there's hydro. What is hydro? Well, it's water again. And then the suffix lysis. Sometimes you'll see L-Y-S at the beginning of a word. Anytime you see that, it means to break something. It comes from a Greek word which means to break something. So in this case, we're going to use water to break something. And so let's take the same molecule that we just had, the sucrose molecule right here, and we're going to add water to it, and we're going to break it back into its individual pieces, glucose and fructose. That is hydrolysis. Dehydration synthesis and hydrolysis work together in your body to make and break macromolecules. This was with sugars, but it works that same way with proteins, with nucleic acids, and with lipids. So let's talk about the 4 major macromolecules, or the 4 types of macromolecules. The first one's carbohydrates. Another word for carbohydrates is sugars. Carbohydrates have gotten a bad rap in today's dietary world, but they're not bad for you. Their function, all these macromolecules are going to have a function. Their primary function is they are the primary source of energy for all living things. They're the primary source of energy for all living things. Most living things will go to carbohydrates and will use carbohydrates more readily than they will use anything else. What are the monomer and what are the monomers and polymers of carbohydrates? Well, the monomer is called a monosaccharide. Anytime you see this word saccharide, it has to do with sugars. So mono means one. It's one saccharide, one sugar. This is glucose. There are other monosaccharides. Here's a disaccharide. Di means two. And here's a polysaccharide. Poly means many. Saccharide, again, sugar. So these are examples of polymers of a carbohydrate. So monosaccharide, polysaccharide are the words you need to know there. Carbohydrates are made up of carbon, hydrogen, and oxygen. And they always exist in a, or not always, but most of the time they exist in a regular pattern. Whereas the amount of carbon is equal to the amount of oxygen, and the hydrogen is the number of carbons times 2. And so you see this in this formula here. This is a glucose formula. And so you have C6H12O6. These two are equal, and this is the carbon times 2. Very regular pattern. Now, carbohydrates can be stored in your body and in plants and other things. When they're stored in plants, it's called starch. They store these long chains of glucose, and they're called starch. Like a potato has lots of starch in it because that's the storage vessel of the potato plant. Glycogen, which is essentially the same thing, is the storage of carbohydrates in animals. And then cellulose, which is a little bit different chemically, is what cell walls are made of in plants. A couple other carbohydrates found in nature. One of them is called chitin. Chitin. Resist the urge to say chitin. It is chitin. And chitin is a carbohydrate that makes up the shells of arthropods, and it also makes up the cell walls in fungi. So, moving on to lipids. Lipids are things like fats, oils, waxes, and steroids. Lipids are extremely nonpolar, so this means that they are also hydrophobic. They're afraid of water. You see the waxy covering of these plants causes water to bead up because the water doesn't want anything to do with the wax, so it's trying to get away. Lipids are generally composed of long chains of hydrocarbons, just carbon and hydrogen, over and over again. There's some other parts of the lipid that aren't simply composed of these two, but mostly the lipid is hydrocarbon. The typical lipid looks like this. The monomer for a lipid is called a fatty acid. And these individual chains here are fatty acid chains. You can see the carbons and the hydrogens. And those fatty acid chains are connected by another molecule up here called a glycerol. Right? Glycerol. And so here is an example of a polymer of a lipid because it's more than one monomer, and it's a triglyceride because it is 3 fatty acids. Triglyceride. And as it were, in animals, these fats can be stored up in tissue called adipose tissue, which is just fat, and fat cells are made. There are 2 kinds of lipids that we're going to look at. Oh, the major function of lipids, I didn't mention that, is that lipids' primary function, as we mentioned kind of up here with the fat, is to store energy. An organism has too much energy, that organism will then store that away. So like an animal in the wild likes to get as much energy as it can. It will have fat stores for the winter or so forth, like a bear before hibernation. There are two kinds of lipids found naturally. They're saturated and unsaturated fats. The difference is that a saturated fat is completely saturated with hydrogen, whereas an unsaturated fat has room for more hydrogen. How do we know? Well, let's look. So notice here on this saturated fat, every single carbon is as full as it can get with hydrogen. There are no more spots left for hydrogen. It is completely saturated. Whereas in this unsaturated fat, notice what you have going on here. You have this double bond between those two carbons that does a couple of things. There are some slots. If we were to undo that double bond, we could put some more hydrogens. But it also causes the fat to kink, or to have this little bend in it, and so you'll see that sometimes on like a structural diagram of a lipid. There are two types of unsaturated fats, mono and poly, whereas mono has one double bond, and poly has more than one double bond. All right, now we can, and saturated fats, real quick, saturated fats are solids at room temperatures, like animal fats typically are saturated fats, and unsaturated fats are liquids at room temperature. Now, why does that have to do with a Twinkie? Well, the fat in Twinkies is actually unsaturated fat, but it has been hydrogenated, meaning that hydrogen has been added to it in order to make those double bonds, single bonds, and artificially saturating the fat, therefore artificially making it solid at room temperature. Another example of a lipid is called a phospholipid. These are these are what your cell membranes are composed of, and here you see again the glycerol fatty acid chains. Notice the kink there because it's an unsaturated fat. And then up here you have this phosphate group, and this is why it's called a phospholipid. In the phospholipid, you have a hydrophobic tail, see the lipids there, and a hydrophilic or water-loving head, and that will dictate the structure of the phospholipid membrane, which we'll talk about when we get to cells. A couple of examples of lipids. There's wax, of course. Wax is basically fat plus alcohol and makes things like lipstick. And then there are steroids. Steroids are a natural thing. They're a good thing. Your body needs them. It's necessary for survival. But steroids can be taken artificially as well, as is the case with this particular ball player. And here's a picture of of some steroids. There's cholesterol as an example of a steroid that your body needs. You can't have too much of that, and it's usually a genetic disorder that causes you to produce too much of a particular kind of steroid. All right, so next is proteins. There's some protein. Here's a picture of the monomer for proteins. This is one of the most important things you learn this year. The monomer for proteins is called amino acids. So we heard them called the building blocks of life before. I guess that's right partially because proteins are very important for life, and amino acids make up proteins. And so the polymer of a protein is called a polypeptide, which is just another word for a protein. This picture of amino acids you will become more familiar with in the coming weeks. Now, one thing you need to know about proteins. Proteins are coded for on your DNA. And so this is called a codon chart. We'll learn to use this in the DNA unit. But each one of your base pairs in your DNA, all the A's, G's, and T's and C's, the U's are found in RNA, ultimately code for proteins. And proteins essentially do everything in your body. So when we start to talk about functions of proteins, well, here's the example of the uh, dehydration synthesis reaction, pulling out water to join two amino acids together, and it forms this bond called a peptide bond. That's why it's called a polypeptide when you have more than one of these together. And here's a protein, just a standard protein. And proteins have many, many functions. And the way that they function is that proteins have a particular shape, and that shape dictates how they are going to work. It's very similar to a tool. When you go to tighten a bolt, you don't go and grab a knife to do that. You go and get a tool that's particularly shaped right for that bolt. And so proteins have a shape that dictate their function. And so here are the 7 functions of proteins that we're going to talk about. Structural, meaning they're used to build things. Signaling, your body uses them as signals like hormones. There's a good example of that. Catalysts, these speed up reactions. We'll talk about that next. Immunity in your body. Regulation of body processes in general. Some animals and plants use them as venom. And then transport of materials across the cell membrane. So proteins have many and varied functions. The proteins have also a particular structure, and there are 4 levels of that structure. This is called the primary structure of a protein. You can write out the word primary if you wish, or I'll do it in the shorthand where you just put a 1 in a little circle above the 1. It's primary. And a primary structure of a protein is just a string of amino acids. These are, think Mardi Gras beads. These are all individual amino acids, and they're all strung together by those peptide bonds that are in there. And this is the primary structure. But when that individual primary structure coils on itself, it makes a secondary structure, and the secondary structure can form either what you see here, like this is called an alpha helix, or it can form something called a beta pleated sheet, or just a beta sheet, which looks like this. And so what happens is you might have, here's an alpha helix, and then you might have a beta pleated sheet here, and then another alpha helix. And so what's happening is all of these individual secondary structures are all binding together. And when they bind together, they will make a particular shape. And that shape is this is the secondary structure. And the next one is called a tertiary structure. We'll go back to this picture. The tertiary structure, for a lot of proteins, this is the completed work. This piece here can actually go off and do some sort of work. But some proteins need multiple tertiary structures in order to function. And this is where you get into what's called a quaternary structure. An example of a protein that has a quaternary structure is called hemoglobin. In your blood, there is a protein called hemoglobin that is made up of 4 tertiary structures, and when they bind together, that forms the finished protein, and that finished protein goes and does a particular job. Now, one of the ways that proteins can be destroyed is called denaturing of the protein. If you've cooked eggs before, you've seen this happen. When you open up an egg, there's the yellow part, that's like the yolk, the fat, and then the white is called the albumin, which is a protein. Well, if you put that protein onto heat, all of those proteins will seize up, and they will all the bonds that were connecting them will kind of break and get all wound up together, and it will cause those proteins to seize up, and that's what makes them taste so good. You can do a similar kind of thing with bleach, because it's a very high pH, or acid, very low pH. In some cultures they cook fish with like lemon juice or lime juice. It's called ceviche. The reason it cooks is because it actually denatures the proteins in that fish. It causes them to cook, very similar to temperature. You can do that with pH, and you can also do that with salt. because you've heard of preserving meat with salt before. And the fourth nucleic acid is called, or the fourth macromolecule is called nucleic acids. And nucleic acids are your DNA and RNA. Function of nucleic acids is to store information, to store hereditary information. Nucleic acids are made up of individual monomers called nucleotides. And a nucleotide is made up of three components: the phosphate group here, a 5-carbon sugar, and this 5-carbon sugar is where the nucleic acid will get its name. It's either going to be deoxyribose or ribose for DNA and RNA respectively. And then lastly is the nitrogenous base. And this is where the nucleotide gets its name, like adenine, guanine, thymine. And so here's a picture of those nucleotides joined together. And so the polymer is just the actual nucleic acid. And again, the function is to store hereditary information. All the code for how your body will work is found on your DNA. And how does your body work? Well, we've talked about it earlier with proteins. And so attached in free earlobes is an example of this. When you were developing, your body had particular instructions to make you with attached or free earlobes, and there were proteins that went out and carried out that function to make your ears a particular shape. This goes for your eye color, this goes for your hair color, how tall you are, and so forth. Every bit of the information about you is stored on your DNA. When we do our genetics unit, we will talk more about how that works.